Puzzling at the time was the observation that these two RNA chains were of constant sizes. If these chains were the actual templates for protein synthesis, we would have expected them to vary in length in relation to the different sizes of their protein products. Equally disturbing, these chains proved very stable metabolically. Once synthesized, they did not break down. Yet experiments at the Institut Pasteur in Paris suggested that many templates for bacterial protein synthesis were short-lived. Even stranger, the sequences of the bases in the two ribosomal RNA chains showed no correlation to sequences of bases along the respective chromosomal DNA molecules. Resolution of these paradoxes came in 1960 with discovery of a third form of RNA, messenger RNA. This was to prove the true template for protein synthesis. Experiments done in my lab at Harvard and at both Caltech and Cambridge by Matt Messelson, Francois Jacob, and Sidney Brenner showed that ribosomes were in effect molecular factories. Messenger RNA passed between the two ribosomal subunits like ticker tape being fed into an old-fashioned computer. Transfer RNAs, each with its amino acid, attached to the messenger RNA in the ribosome so that the amino acids were appropriately ordered before being chemically linked to form polypeptide chains. Still unclear was the genetic code, the rules for translating a nucleic acid sequence into an ordered polypeptide sequence. In a 1956 RNA Thai Club manuscript, Sidney Brenner laid out the theoretical issues. In essence, they boiled down to this: how could the code specify which one of 20 amino acids was to be incorporated into a protein chain at a particular point when there are only 4 DNA letters, A, T, G, C? Well, obviously, a single nucleotide with only 4 possible identities was insufficient, and even 2, which would allow for 16, 4 times 4, possible permutations, wouldn't work. It would take a minimum 3 nucleotides, a triplet, to code for a single amino acid. But this also supposed a puzzling redundant capacity. With a triplet, there could exist 64 permutations, 4 times 4 times 4. Since the code needed only 20, was it the case that most amino acids could be encoded by more than one triplet? Now, if that were so, in principle, a quadruplet code, 4 times 4 times 4 times 4, yielding 256 permutations, was also perfectly feasible, though it implied even greater redundancy. In 1961 at Cambridge University, Brenner and Crick did the definitive experiment that demonstrated that the code was triplet-based. By a clever use of chemical mutagens, they were able to delete or insert DNA base pairs. They found that inserting or deleting a single base pair results in a harmful frame shift because the entire code beyond the site of the mutation is scrambled. Imagine a 3-letter word code as follows: Jim ate the fat cat. Now imagine that the first T is deleted. If we are to preserve the 3-letter word structure of the sentence, we have Jim ate hef atk at, gibberish, beyond the site of the deletion. The same thing happens when 2 base pairs are deleted or inserted. Removing the first T and E, we get Jim, ath, effa, tk, t. More gibberish. Now, what happens if we delete or insert 3 letters? Remove the first A, T, and E. We get Jim the fat cat. Although we've lost one word, ate, we have nevertheless retained the sense of the rest of the sentence. And even if our deletion straddles words, Say we delete the first T and E and the second T, we still lose only those two words, and are again able to recover the intended sentence beyond them. Jim, ahi, fat, cat. So it is with DNA sequence. A single insertion deletion massively disrupts the protein because of the frameshift effect, which changes every single amino acid beyond the insertion deletion point. So does a double insertion deletion. But a triple insertion deletion along a DNA molecule will not necessarily have a catastrophic effect. They will eliminate one amino acid, but this does not necessarily disrupt all biological activity. Crick came into the lab one night with his colleague Leslie Barnett to check on the final result of the triple deletion experiment and realized at once the significance of the result, telling Barnett, "We're the only two who know it's a triplet code." With me, Crick had been the first to glimpse the double helical secret of life. Now he was the first to know for sure that the secret is written in 3-letter words. So the code came in threes, and the links from DNA to protein were RNA mediated. But we still had to crack the code. What pair of amino acids was specified by a stretch of DNA with, say, sequence ATA, TAT, or GGT, CAT? The first glimpse of the solution came in a talk by Marshall Nirenberg at the International Congress of Biochemistry in Moscow in 1961. After hearing about the discovery of messenger RNA, Nirenberg, working at the U.S. National Institutes of Health, wondered whether RNA, synthesized in vitro, would work as well as the naturally occurring messenger form when it came to protein synthesis in cell-free systems. To find out, he used RNA tailored according to procedures developed at New York University 6 years earlier by the French biochemist Marianne Grunberg-Monango. She had discovered an RNA-specific enzyme that could produce strings like AAA, AAA, or GGG, GGG. And because one key chemical difference between RNA and DNA is RNA's substitution of uracil, U, for thymine, T. This enzyme would also produce strings of U, U, and so on, poly-U, in the biochemical jargon. It was poly-U that Nirenberg and his German collaborator, Heinrich Mattei, added to their cell-free system on May 22, 1961. The result was striking. The ribosomes started to pump out a simple protein, one consisting of a string of a single amino acid, phenylalanine. They had discovered that poly-U encodes polyphenylalanine. Therefore, one of the 3-letter words by which the genetic code specified phenylalanine had to be